All right. So, today's discussion session is about an organism. Recently isolated, so recently in fact that the paper hasn't been published yet, hasn't even been peer-reviewed yet. But a lot of people these days will post preprints on preprint servers around the world so that people can have a look at them. I think you can argue that it even helps the peer review process. This paper is by a Japanese group, Imachi and Nubo, and then there's a whole string of other authors. And the reason there's a whole string of other authors is because this was a monumental effort spanning 12 years' time to cultivate this organism. And so if you think that 5 or 6 weeks in microbiology diversity lab is a long time to struggle getting an organism in pure culture, you haven't seen anything. The presumptive title of this paper is Isolation of an Archaeon at the Eukaryote-Prokaryote Interface. And this is a scanning EM of this picture. We'll see this picture and some others in some detail. It's a monogram. So a monogram is a formal description of a new species. And it's in a monogram that you give an organism a formal name. You also have to describe various aspects of its phenotype. These days, I think it's almost a prerequisite that you have to sequence its genome instead of some of the old-fashioned stuff you have to do. And it's a pretty interesting organism. And so you might ask yourself, why 12 years ago would you set out on an effort that you knew was going to be hard? They certainly knew that. And 6 years down the road, you haven't got it yet, but you're still willing to work at it. And 2 years after that, you still haven't got it yet, and you're still willing to work at it. Why on earth would you be willing to do that? The reason is that this is a really, really interesting group of organisms. It's a large group of organisms for which there are no cultivated members before this paper. And so this is a phylogenetic tree of the archaea. And so if we go back to our universal three-domain trees, here's the root. The bacteria head off that way. The eukaryotes head off that way. And there are, by some ways of thinking, 4 major groups of archaea. There are actually 2 major ones. The Euryarchaea. These include methane-producing organisms, methanogens. That includes the halophilic archaeas, things like Phylogenetic. There's what some people call the TAC group, or others would call the Crinarchaea, that are the typical sulfur metabolizing thermophiles, things you see in hot springs and that sort of thing. There's a group of environmental organisms. These are organisms, again, nothing in that group has ever been cultivated, so we don't know as much about them. But their sequences have been fished out of DNA samples from all over the world, primarily sediments and things. And then there's this group here. They call them the Asgard archaea. People run out of names after a while, right? And in the world of archaea, historically, there's been a lot of Germans and Germanic folks involved. The German government, after the discovery of the archaea, dumped a ton of money into isolating new, weird, extremophilic organisms. And so a lot of the work on isolating and cultivating archaea comes from Germany and other Germanic countries. And so, you've got the Norse god thing going on with this group of organisms. One of these groups is the Loki archaea. How many of you ever heard of the Norse god Loki? Yeah, apparently it's some movie now. Anyway. As someone with some Icelandic ancestry, this is a little befuddling. So anyway, the interesting thing about these Asgard archaea, and particularly the Loki archaea, is that folks went into an environment where there were some Loki archaea, and they fished DNA out, and they were doing metagenomics, that is, just sequencing all the DNA they could get and trying to assemble genomes to see what kind of organisms these were. And when they did that, one of the assembled genomes was from a Loki archaeon, but it contained eukaryotic genes. So genes for actin, genes for tubulin, genes for G-coupled receptors, genes for cell cycle regulation. And so they become very interesting, right? In fact, one of the things that's happened recently is that although this tree with the eukaryotes branching off here is what you see with ribosomal RNA tree analysis, and people have done this now for 30 years or more, 40 years or more. It turns out that as people try to do more genomic-based trees with ribosomal proteins instead, because these are small proteins that are easy to identify, easy to align, etc., the trees come out with the eukaryotes coming out here, out of the Prometheoarchaea, and in particular, probably with the so-called Asgardarchaea. Now, I can point you to a paper that shows you exactly why this is an artifact of their treeing algorithms. Of course, the folks who do that would argue otherwise. And so I think anyone who says with certainty exactly where the eukaryotes and how the eukaryotes and archaea are related, if they're absolutely convinced, then they're fooling themselves. I think there's a lot of uncertainty in that. And I think that's interesting. But regardless, here we've got a group of archaea that may or may not be specifically related to eukaryotes with what appear to be some eukaryotic genes in them. By the way, if you believe the ribosomal protein trees, then this is the tree you get. If you believe the ribosomal RNA trees, then maybe this branch is further down here and actually over on this side instead. And so the Asgardarchaea wouldn't formally be archaea, but actually a primitive form of eukaryote. But when you take sequences out of the environment and you assemble fragmentary sequences to try to create a genome, there's always the danger that you're putting apples and oranges together in the same genome. Could those eukaryotic-like genes have come from some other organism and the assembly process mashed them together as an artifact? But if those organisms do contain eukaryotic proteins, if they are specifically related to our eukaryotic ancestry, might they have some clues to how eukaryotes originated? Wouldn't that be really interesting to look at them? And in particular, when you think about the ancestor of the eukaryote, according to, let me see if this is the next page. Yeah, according to the traditional ways of thinking about how eukaryotes work, you have either an archaeon or some proto-eukaryote and an endosymbiosis. And usually people think, because it's absolutely clear that mitochondria, Golgi apparatus, nuclear envelopes, etc., did not come from the alpha proteobacteria that was a symbiont that created the mitochondria. And so they must have come from somewhere else, right? And one possibility is the archaeon or proto-eukaryote that engulfed that alpha proteobacterium to create the last eukaryotic common ancestor. Another possibility, of course, is that an archaeon or a proto-eukaryote, which had a simple cell structure, what you might call a prokaryotic cell structure, formed a symbiosis with this alpha proteobacterium and then developed all those eukaryotic specific cellular structures. But if you're going to look for this, what you're looking for is a primitive organism deeply, deeply related to the eukaryotes or archaea that has proteins for cell cycle regulation and tubulin and actin and all that kind of stuff. And here's a candidate for that, right? The Lokiarchaea. This is why they were so interested in isolating this organism. "12 years. The first step was to go down to this deep sea hydrothermal vent where Lokiarchaea had been discovered in a deep sea submersible and take some samples. This is a methane seep. Do you guys know what a methane seep is? Where you have oil deposits, particularly, that will seep methane, natural gas, right? But at the temperatures and pressures of the deep sea, that's not a gas. When it mixes with water, it forms a hydrate, methane hydrate, which is a solid material. And that's what methane seeps are made of. And that methane slowly bubbles away out of the methane seep. And methane oxidizers in the ocean oxidize that methane. And so this is a methane seep. They started a primary enrichment, and they ran that for 5.5 years. And so they recreated this kind of environment in the lab, in a bioreactor. They pumped methane into it and just let stuff grow. They were monitoring it by fishing bits of DNA out and looking for Lokiarchaeal sequences, right? And so when they started to see those accumulate, they did a second enrichment in tubes. And they didn't do this blind. They believed from the partial genome assembly that there were certain amino acids that this thing probably couldn't make. They believed it was because it's a methane seep that it somehow is using methane, and so they're pumping methane in. And after a year, those tubes started to get a little bit turbid. And so they looked at it. And what they found was that most of the organism there was a fairly mundane proteobacterium, Halodesulfa vibrio. It's a sulfate reducer. This is an organism that takes organic material and goes through standard respiration using sulfate instead of oxygen as a terminal electron acceptor. But they also found some Lokiarchaea. And so they subcultured this mixture 6 times, each time waiting until a fraction of Lokiarchaea was as high as it was going to get, and then transferring it again. And so trying to get it built up as an enrichment with the most amount of Lokiarchaea in it. And at the end, they had 13% compared to 85% of Halodesulfa vibrio, and a little bit of this methanogen, methanogenium. They found this interesting, and this one as well, because one of the things both of these organisms can do is take electrons from other organisms and use them to make a living, either by sulfate respiration or by methanogenesis. It turns out that organisms that make hydrogen for a living, and the Lokiarchaeon looked like it might be such an organism. It looked like it might carry out kind of standard fermentation. And whenever you do a fermentation, the problem is, what do you do with the electrons left over? And so you're taking some organic compound, you're oxidizing it, you've got to put the reduction somewhere. And a lot of fermenters will dump them on other organic compounds and make things like ethanol or lactate or whatever. Some organisms have a hydrogenase, and they just dump the electrons onto protons and make hydrogen. Well, I'm sure you can understand that making hydrogen is not usually an energetically favorable thing to do. And so in order for hydrogen production to be favorable in microbiology, the trick is you have to keep the concentration of hydrogen low as you can. Really, 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 really low. And the way this is done in most biological systems is that hydrogen generators live in close symbiosis with hydrogen consumers. And so they trade that hydrogen off. The hydrogen consumer does something with it to get rid of it, and now the organism can continue to make a living generating hydrogen. And so, for example, if this organism, which they're going to call Prometheoarchaeum syntrophicum, is living in symbiosis with methanogen, the methanogen will take that hydrogen and CO2 and make methane for a living and get rid of the hydrogen so that the Lokiarchaeon can continue to prosper. So they did a bunch of other things, and after a while, they got a pure co-culture with just these 2 organisms in it. The Lokiarchaeon, which they want to call Prometheoarchaeum syntrophicum, and methanogenium. 12 years after collecting their inoculum is how long that took. Well, they sequenced the genome of it. They did some fluorescent in situ hybridization of these co-cultures to find out what they look like and all this kind of stuff. They did stable isotope probing to figure out who's providing what nutrients and that sort of thing. We'll talk about these technologies later in the semester. They did a thing called nanosims, nano-stable isotope mass spectroscopy. They did some electron microscopy, some basic biochemical and microbiological tests. And they're just trying to figure out what this symbiosis is about and what the organism Prometheoarchaeum is like. So this is what it looks like. It's an anaerobic mesophilic heterotroph. It grows optimally at 20 degrees. It's a heterotroph, and it's an anaerobe. It hydrolyzes peptides and oligopeptides into amino acids, or it can eat amino acids directly, and it oxidizes those. And when it oxidizes them, it makes formate, which it can excrete into the media as a waste product, and it makes hydrogen. And again, it's got to get rid of that hydrogen in order to prosper. It also has some amino acid and vitamin requirements. That is, there are some amino acids and things that it can't make, and it's presumably getting those either from the medium or from this symbiosis. This is quite common, of course. I'm sure you're all aware that there are 11 amino acids that you can't make. We call these essential amino acids, right? Because we have to get them in our diet. And likewise, there are some amino acids that this can't make. It is promiscuous. This is not a judgment. What this means is that it can live in symbiosis with a number of different kinds of organisms. They've tried 3 of them. Of course, they isolated it as a symbiontum methanogenium. They can also grow it with just halodesulfovibrio, and they can also grow it just with a completely separate organism that they pulled out of their culture collection, Methanobacterium. And so all it needs is some amino acids and vitamins and somewhere to dump the hydrogen. Now this organism is interesting because it produces a bunch of blebs. These are all blebs, all this stuff, and appendages. And this is not so unusual in the microbial world. There are lots of microbes with appendages. Many of you who are in the lab have rhodomicrobium in your purple non-sulfur tube, which is appendage. It either has stalks or fibers that connect one cell to the other. They don't say so in the paper, but they had to be hugely, hugely disappointed when they looked at this organism. Because it's got no nuclear envelope, it's got no Golgi apparatus, it's got none of this typical eukaryotic cell structure stuff. But the genome does contain those genes, those eukaryotic-like genes. And so that was not an assembly artifact, but it actually does have G-coupled receptors and GTPases, it has actin and tubulin. That's pretty interesting. Now, I would be very interested myself when this paper actually comes out, is to fish those sequences out and see how similar they really are to actin and tubulin, and see whether they might be a recent horizontal transfer into the organism or whether they might be primitive versions of those proteins. How many GTPases and G receptors and things are there? Cell cycle regulators. Here's some more pictures of it. It seems to lose the appendages when it goes through division, which it does about every 20 days. This is really a slow-growing organism. And so you see this is going through 2 rounds of division at once, and you can see that the appendages are essentially gone. There's a bit of one up there with the arrow. It's still producing some blebs. Interesting to me, it's producing blebs here at the old division plane, and there might be some cell biology there that's worth pursuing. Notice up here at the top, one way in which this organism is different from other bacteria, well, from bacteria that form appendages, or archaea that form appendages for that matter, is that these appendages are complicated. Here's an appendage and it's got a fork in it. Here's another forked one. And they're producing blebs and all this kind of stuff. So here's a beginning of an appendage. So these are pretty complicated appendages. Now there are other archaea that have really thin fibers, cytoplasmic fibers that connect one cell to another. Parabaculum comes to mind. We'll show you some pictures of that later on. But these are pretty robust fibers. These are clearly cellular structures. I mean, they've got an envelope around them, a cytoplasm, and a volume inside to do something with. And these cells are small. So this is a micron. The average size of the kind of irregular coccus in the middle is about half a micron. So these are right on the edge of being visible by a light microscope. These are not the big, complicated proto-eukaryotes they were looking for in the first place. And this is a bunch of them all massed together. They like to grow in clumps, and so you get these big masses of cells, and you see the connectors, the appendages here, and blebs, and all that kind of stuff. Here's some more pictures. This is a cross-section, a thin section, stained with heavy metals and stuff. And what you can see, I mean, it's got a typical archaeal membrane with an S layer on the outside, protein semi-crystalline outer coat. It's got a nucleoid in it. You can't really see it that well in here, but there's no envelope around that nucleoid. There's no internal cellular structures, membranous structures, no eukaryotic-like structure here at all. And here's one that's fairly recently divided, and it's beginning to produce some appendages here. So, you spent 12 years trying to isolate a proto-eukaryote, and you get an archaeon instead. What do you do? Well, this is a pretty interesting organism. I mean, number one, you'd really like to look and start to do some experiments and figure out what that actin is doing and what that tubulin is doing and how its cell cycle works and all that kind of stuff. Because those things are relevant to eukaryotic, the relationship between this organism and eukaryotes. The problem, of course, is that this organism is so hard to grow and grows so slowly and to such low cell densities. You know, if I were going to do a tubulin assay, I would want to grow, you know, at least a couple of 100 milligrams of cell paste. And that would be a real task with this organism. So doing experiments with organisms like this is really hard. But I'm sure they're underway. So what they do is they propose a model. They're interested in this organism as a proto-eukaryote. That is this kind of an organism similar to something that might have ultimately been our ancestors and evolved into more complicated cell structures. And so they propose a model, and it's an interesting model, and I doubt that it's correct, but it at least it's a way of thinking about it, and I kind of like. So here's the organism itself, and all the fibers and things. And it takes in amino acids, and it excretes organic acids, formate, acetate, propionate, butyrate, and it exchanges some stuff with this other organism. But primarily it's giving it hydrogen, which this organism makes methane from. This is methanogenium they're showing here. And so it's a great symbiosis. Now, mitochondria did not descend from methanogens. They descended from alpha proteobacteria. So what they're going to argue is that it produced a symbiosis also with an alpha proteobacterium. And in this case now, instead of the methanogen, they're showing the sulfate reducer as the other organism in this symbiosis. And so both of these organisms are making hydrogen, but what this is providing is a way to fix carbon, because this might be an autotroph, and could trade things back and forth. But this is the key. In early Earth, until about 2.2 billion years ago, the Earth had a really, really, really low oxygen concentration in the atmosphere. About the time eukaryotes may have originated, and this is a bone of some contention, all the ocean buffering capacity for oxygen was depleted. And oxygen started to accumulate in the atmosphere. And so organisms, microbes that lived in the world at that time had to deal with this oxygen catastrophe. And what they're proposing is that the ancestor of eukaryotes did this by forming a symbiosis with an aerobic organism. And the trick is that this organism is sucking up oxygen in the immediate vicinity and keeping the oxygen concentration low. Either this one or certainly this one are generating hydrogen, and the sulfate reducer has to be there. Sulfate reducers are quite common in ocean environments, which is why they kind of like, I think, that one instead of methanogen. Methanogens aren't able to live even at the lowest oxygen concentrations. And so if there's enough oxygen for this to live, you can't have a symbiosis with a methanogen. So they like the sulfate reducer instead. As the oxygen concentration goes up, it's got to form an even more intimate relationship with this oxygen scavenger. And so it engulfs it, so to speak, with blebs and fibers, making sure all the oxygen goes to it, and it keeps the oxygen concentration in that vicinity low. This is not outrageous. There are organisms that do this kind of thing. There's pretty good reason to believe, for example, that rubisco, the gene for fixing carbon in the Calvin cycle, rubiscos bind CO2, right? But they also bind oxygen. In plants, we call this photorespiration, if you think back to that. No one has ever been able to fix rubisco so that it doesn't do photorespiration. Plants go through all kinds of gyrations to keep oxygen away from rubisco. Rubisco may have originated as an oxygen scavenging protein rather than as a carbon fixing enzyme. And so this Lokiarchaeon or proto-eukaryote, which may be related, became more and more intimately symbiosed with this oxygen scavenger, and just eventually just filled in the gaps and completely engulfed it. And now you have this alpha proteobacterium that runs aerobic respiration, does amino acid stuff. Doesn't that look familiar? That's what mitochondria do. Your nuclear cytoplasmic lineage doesn't do any respiration. It's an anaerobic system. It's only the mitochondria that does that kind of stuff. But you're still passing off that hydrogen to a sulfate reducer. And how you deal with that, I think, is one of the open questions in this model. Is, still what about that hydrogen? This is really, really similar to an older model, and I put a link to this paper on the Moodle page. It's called the inside-out model for eukaryotic, for the origin of eukaryotic cells. The typical one is called the engulf and enslave model, where some proto-eukaryote picked up the alpha proteobacteria by phagocytosis, and so it engulfed it and it enslaved it. The authors of this paper are calling it, in their model, the entangle, engulf, and enslave model. You'll notice when you look at this paper that they really like their acronyms and their cute little labels for things. I find this incredibly annoying. But Baum and Baum put this model together a few years ago called the inside-out model, and the notion here is pretty similar. And so you've got a symbiosis of some kind. An early eukaryote, they're calling this the eocyte. It turns out that the notion that archaea may have been the origin of eukaryotes has been around for a very, very long time. There was a guy named Jim Lake, when the Three Kingdom thing first came out, who argued, based on some particular phylogenetic trees that had their own problems, that eukaryotes evolved from Crenarchaea. And he called this the eocyte model. And that's where this word eocyte comes from. And so it forms a symbiosis with alpha proteobacterium. It forms pseudopods of some kind. And this looks familiar, right? This is what Prometheoarchaeum seems to do. And ultimately, more or less engulf them. And in this model, the Golgi and the endoplasmic reticulum and all this internal membranous stuff is the remnant, now repurposed for other things, of this whole engulfment process. I think that's a pretty interesting model. It leaves a lot of things out. Let me give you an example. where in this system does this organism here make its proteins? Presumably in here, right? That's the cell. And so today, you don't make proteins here. You make proteins out here. So there's some things like that need to be sorted out. But it's an interesting model. And so I think the authors of this paper can reasonably argue that they've isolated an organism that's a candidate for this sort of a model for the origin of eukaryotes. This is a marine tree, a tree of marine microbes, I don't even remember where I picked it up from. You're not supposed to do that, right? But I thought it was interesting because it shows the bacteria here, it shows archaea here and eukaryotes here, and you can argue that the way they've done this tree, although they've colorized this part, this cyan color, that is that these eukaryotes come from archaea, you could leave that uncertain, right? And now the archaea are here, eukaryotes are here, and in that tree, the loci-archaea are primitive members of the eukaryotes. And I think this is a reasonable way to think about it. I think that if you can't resolve what that particular deep branching pattern is, maybe it doesn't matter so much. I mean, it's a phylogenetically really interesting question. But in terms of what it means in terms of the models of the origin of eukaryotes, it's not clear to me how important that is. Your task is to look through this paper, go through the presentation. I put a couple of other links that you'll find interesting. I put a link to that inside-out model paper. I put a link to a paper that argues very strongly that the eocyte tree, that is the trees where the eukaryotes come out of the archaea, is probably wrong. There's a whole bunch of public reviews of this paper that are out there. Nature, the journal, has one. Science has one. Science Direct has one. Just do a search and you'll find all kinds of stuff. You'll find actually that much of the text is the same from one to the other. They borrow pretty freely. Find something that you find interesting. I mean, if you can't find the origin of eukaryotes interesting in any way, then I don't know what to think. Find something interesting about this organism, about how they isolated it, about their model for the origin of eukaryotes, and make a primary post, a discussion. Look stuff up, all right? I'm asking you to dive into it. Look into it deeper. Go to the literature, do some searches, find out what people know or what they think or what they used to think or whatever, and post what you find and maybe what you think about it. That's due by Friday midnight, and then over the weekend, as usual, take a look at all the other stuff that people have posted and post a couple of comments, including the possibility of comments on your own work posted after you've seen what other people said about it. Yeah?
Today is Friday, right?
Today is Friday. Yeah. Any questions about this? All right, have fun with it.
I need to go take